Oh. I'm on here just waiting for some of y'all to join. I'm exhausted. I'll just sit here and stare at the screen. As some of y'all go to look for the link and join. Whenever you get on, go ahead and say, hey, 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 Jacob, what up? All right. Okay. Who's Vapor and Flare and Jolt? And dear Lord, people. Okay. As you're on, hey, I can jump into anything. Oh, Heather, hey. Okay, I can jump into whatever you need me to jump into, or it'd be best if y'all actually just comment and say what you want me to cover. Like, you want me to do cell cycle, you want me to do pictures, diagrams, pedigree, something like that. So it'd be best if you just like comment. If you're having issues seeing the comment thing, it's probably because you just need to open up the window all the way. Are they going to ask about independent, dependent? No. I don't think I've ever seen them ask independent or dependent. They've never done those. Okay. All right. So real quick, Katie, can we go for pedigrees? Yes. Do we need to talk about the Krebs cycle in detail? Not necessarily, but I'll get to that, Jacob. All right. Pedigree. Okay. So all I have is this green marker. So hopefully it shows up well enough for you. Okay. So first thing in a pedigree that they would probably end up asking would be something along the lines of, how many generations are there? So generations get numbered from top to bottom. So only 2 generations. Individual, let me see. Let me do this. Generation 2, individual 2. Generation 2, individual 2. Does this person have the trait? Well, generation 2, and you always just count straight across. So 1, 2. The fact that it's not shaded tells you no, they do not have the actual trait, right? That would be kind of a basic question. Another question would be, how many offspring does this couple have? 1, 2, 3. This person is married into the family. They're not part of the actual offspring because they don't have a line going to them. Again, also in pedigrees, the fact that I have children shaded and I don't have any parents shaded means that they have to be recessive. That this trait is recessive because the only way that parents wouldn't be shaded is if they have the opposite. So whenever you determine what the shading is, okay, Twitch, lethals, PC, I don't know what any of that means, Michaela, okay? Whenever you determine whatever the shading is, if you said the shading is recessive, then label it. I put homozygous recessive, homozygous recessive. That means everybody else who's not shaded, you need to go ahead and put a dominant. So see if that works out. Can dominant have recessive? Well, yes, if they're heterozygous, they certainly can. But let's say you accidentally did this, okay? Let's say you looked at this shading and said, well, I think that shading's dominant. So you put a capital letter on the shading. That means everybody who's not shaded, you need to put the opposite, recessive. And guys, recessive can only show up if it is homozygous recessive. So that means these parents are homozygous recessive. They have no dominant A that they could even give to their offspring. So you know you did it wrong. Okay, so you need to kind of flip that. All right. Katie, I don't know that there's much more that we need to go into detail for pedigrees, but if you want me to do another one, I certainly can. You just need to let me know. Okay, Jacob, as far as the Krebs cycle, not so much detail as a couple of key points. So if you're talking the cell, okay, glucose comes in. Glucose is a 6-carbon molecule, C6. That gets broken down into this guy, pyruvate. Pyruvate was a 3-carbon molecule, okay? That 3-carbon molecule then will be moved into the mitochondria. And in the mitochondria, that pyruvate immediately gets broken down and gets put into the Krebs cycle, right? So pyruvate, acetyl-CoA. Good night, baby. Hey, hold on. Sorry, just a second, my daughter. Do you need to wash your hair? Probably.
No, I did that last night.
In the bathtub? Yeah. With all the oil? Okay. Okay. Sorry, talking to my daughter. She'd be nasty. Okay. Anyways, so in the Krebs cycle, pyruvate will move into the mitochondria because the first thing it's going to do is go to the Krebs cycle. So the Krebs cycle helps to break pyruvate down. And here's the only thing that would be detailed for Krebs cycle, Jacob, is what does the Krebs cycle produce? The Krebs cycle produces CO2. That's the detail that really you would have to know, is it produces the CO2. Why? Because it's the complete opposite of the Calvin cycle in plants. In the Calvin cycle in plants, okay, plants take CO2 in, put it into the Calvin cycle, and make sugar. So they do the opposite of us. So that would just be kind of the key point you'd want to know as far as the Calvin and Krebs cycle. Krebs is for crabs, so that's animals' respiration. Calvin is for plants because the plants in my room are named Calvin. Okay. All right. What's another one? Michaela, I still have no idea what in heaven's name you wrote when you said Twitch Lethals PC. Okay, sounds good. Okay, Cass, can you go over gene flow and all that stuff about genetic drift? All right. So when you're talking any of those things, anything having to do with gene flow or allele frequencies, what you're talking about is evolution. If evolution is going to happen, it's because there's some kind of change in your frequencies of alleles. How often do you see dominant? How often are you seeing recessive? So the only ways that you can change allele frequencies is through a couple of options. First, and here's the thing, all those 5 things fall under the Hardy-Weinberg stuff. Yeah, I know y'all don't want to hear that. There were 5 things that were unique about Hardy-Weinberg. Remember, Hardy-Weinberg was the P and Q stuff. But to even do the math, there were 5 things that the scientists said. Because remember, what they were trying to prove, or in a way disprove, was if evolution is going to happen, then that means these 5 things must always be acting on it. So what they did is said, fine, you don't think evolution happens, then let's go ahead and play your game. If evolution never happens, that means frequency, meaning how often, of dominant and recessive you see can never change. So if you saw dominant 45% of the time in one generation, then for 50 generations after that, you're going to see 45% dominant every time. We know that's not really the case, but they said, fine. But for that to happen, 5 things must always occur. The problem is no real population can ever actually hit those 5 things. So they said, first, you must have a large population. Okay. Well, that means literally every population of dogs, every population of insects, every population of humans must be very large. That means go to every distinct corner of the world. It must be a big population. Doesn't really happen. 2, you are never allowed to have mutations. Okay. You can't control that. Mutations are random. You are never allowed to have natural selection. Again, it's called nature for a reason. You're always interacting with the environment. You're always going to be under some kind of natural selection. 4, no gene flow. All gene flow means is no DNA flowing between populations, meaning no migration. So if you don't have migration, you're not introducing new genes and new DNA, which means your frequencies would always stay the same. Well, that's not really how it goes, right? You have people always moving around, introducing insects, introducing dogs, introducing their genes all the time. And then the last one is, random mating. You are never allowed to select your mate based on good genes or not. Nobody does that. The chicken doesn't want to go with that rooster that's missing half of his feathers. We don't randomly mate. We want the best genes possible, okay? So when it comes to things that can affect allele frequency, gene flow is one of those. Migration. The other one you mentioned was genetic drift. All genetic drift means is by chance. That genes have the chance to drift in and out of frequencies by chance. Genetic drift are the things that happen that you don't always have control over. There are two things. Bottleneck effect. A bottleneck effect means that some kind of natural disaster happened. A flood, a fire, an earthquake, a volcanic eruption, something. Killed half the individuals, you have no idea. Genetic drift can also happen through what we call the founder's effect, where certain individuals, yes, the green ones. Sorry, my daughter again. Where certain individuals get moved and start a new population. So birds pick up yellow flowers, transport those yellow flowers, drop the seeds to another area, but maybe in the original there was yellow, blue, and red. Well, in the new population, there's only yellow. Okay, in a founder effect and bottleneck effect, it's completely and totally random on what you can do. Okay, can we go over the math problem we have to know? Trinity, I'm thinking you are talking about the Hardy-Weinberg again. So yes, I'll do that. Nate, quick protein synthesis explanation. Yes. Do bacteria have nuclei? Cass, no. No bacteria. Remember, bacteria are your examples of prokaryotes. No. That no means they do not have a nucleus, nor do they have organelles. Rachel, can you go over cell transport? Yes. No. Thanks, Nate. All right, so Trinity, the only math you had to know was Hardy-Weinberg. The two variables you have to know is P and the letter Q. If it helps, always write the P as a capital letter and write the Q as a lowercase letter. So the P represents the dominant, the Q represents the recessive. Now, notice how I just wrote them as an individual letter. That's because the individual P and Q represents the allele. But the second that I write P squared, now I'm talking about the genotype dominant. Or Q squared, I'm talking about the recessive genotype. Or 2PQ, notice how it has both letters in it. Now that's hetero. So the equation, remember there are always the same 5 steps in this problem. Always, always. First of all, you can only ever go through the Q squared door. Why? Because Q squared actually means lowercase q, lowercase q. Those are the recessive people. You have to start with these guys because they are the only ones you actually know what they are. Because recessive is truly recessive. Dominant, you don't know. They could be either. So you start with the Q squared door. So I'm just going to make one up. I'll have to do the math kind of right here on my calculator, on my phone, because I don't know the math, square rooting and stuff in my head. Okay. All right. So let's say butterflies, white are 15%. Okay. And white is recessive. Solve for the Hardy-Weinberg for all the genotypes and allele frequencies. Right there, the fact that I gave you the percentage, you can only ever do this math in its decimal form. So I know that that's Q squared, but because I was talking about butterflies. Butterflies are the genotype, not the alleles. Butterflies have both, 2 alleles, genotypes. So that Q squared, I'm going to write as a decimal, 0.15. That's step 1. You just got to write what the problem gives you. Step 2, solve for Q from what you just had. So take that 0.15. Got to, like I said, I ain't doing this math in my head. 0.15 gets you 0.39. Okay. The next thing you do is solve for P. You know all P is P plus Q equals 1. So algebraically, plug in Q, solve for P, which means subtract that by 1, which gets you 0.61. Then step 4, p squared. Okay. Yes, you have a calculator for the test. Okay, p squared. So take that 0.61 and square it. Ooh. So I get 0.37. And the last thing you solve for is 2pq, which means literally take the number 2 times what you found for p and what you found for q. So 2 times 0.61 times 0.39, ooh, equals 0.48. The only last thing you'd want to double check is make sure the p squared, the 2pq, and the q squared all equal 1. It looks like they do, so you're good. The only thing that if they give you a Hardy-Weinberg, because it's very doubtful they actually would, if they were to give you a Hardy-Weinberg, it would ask you for what percent of the population is heterozygous. Hetero would be a P and a Q, so you simply would find 48%. What percentage of the population has the dominant allele? 61%. Okay. If you can remember those same 5 steps, you're good. Okay. Let me go back up a little bit for a second because some of y'all have gone through a little bit. All right, Nate, I'm going to do protein synthesis for you. Then I'll do cell transport, Rachel. Okay, what is a virus doing like HIV when it has all the time before you get affected? There was a question on the packet about it, and I didn't know. Okay, viruses like HIV are lysogenic. They literally inject their DNA, and they hide in your chromosomes, and they just sit there. The DNA does. It just incorporates itself into your chromosomes. DNA is DNA. Your cell lives its life. Because at this point, all it is DNA inside of you, viral DNA. So it hides, it doesn't do anything. Your cell goes about its life. But HIV infects your T cells. That's your immune system. So while your immune system is there, healthy, replicating, going about its life, it doesn't realize, but it's also replicating tons of viral DNA. And then at a certain point, the viral DNA finally gets triggered. Starts replicating, and it kills and breaks out of all of those T cells. Okay, Nate, protein synthesis. That was the transcription translation. There are always the 3 same steps. Transcription to translation. Always kind of starting with DNA replication at the beginning. Now, C comes before L. So in the transcription, you got to write it before you can translate it into your language. You got to write it down before you can translate it. So transcription happens inside the nucleus. That's DNA giving the message to mRNA. So that is DNA saying, I have a TGC or ATGC. mRNA, I need you to copy down UACG. That's what transcription is creating an mRNA molecule. So take the DNA and make an mRNA. There are no trees in the land of RNA, only you. As soon as mRNA is created, single strand, that single strand leaves, finds a ribosome, lays in that ribosome, tRNA, kind of looks like a T. tRNA brings its amino acid. That amino acid starts being left behind. Amino acid, amino acid, amino acid, amino acid. And that amino acids are monomers that get bonded by peptide, and you ultimately start building your protein. That is translation. DNA, mRNA, to your ribosome, which is simply rRNA, meets up with the tRNA, build your protein. If you can kind of have that story in your mind, the Dawn has to talk to Mikey. Mikey goes down to the docks, meets up with Tony. Tony leaves the package. And at the end, you've got all your protein meat necessary. Okay. All right, Rachel, cell transport. Okay. Let me know if y'all have any more questions. I'm kind of just moving quickly because I know some of y'all are getting on, some of y'all are getting off. It's really late. I know I'm exhausted. Some of y'all may still want to study just on your own. All right, so cell transport. Rachel, I think what you're asking for is just passive and active transport. There's only two big categories for transport. Passive simply means no energy is needed, no ATP. The only way passive transport can happen is two methods. First, diffusion. Diffusion is going from high concentration to low. You spray perfume in the corner, it will eventually diffuse across the room because it wants to go from a high concentration of itself to where there's little of itself. Okay? Water is a type of diffusion called osmosis. Only water can move through osmosis, but it is a type of diffusion. Okay? There's only one other kind of passive transport, and that's called facilitated diffusion. You know it's passive transport because it has the word diffusion in it. I understand some of y'all get stuck on the word facilitated, and you're like, what does that mean? It means to help. But the fact that it has diffusion in it tells you it's passive transport automatically. Facilitated diffusion means, here's my cell. My cell has special proteins in the membrane so that things that are too big can actually move through and get into the cell, such as sugar, proteins, lipids. Things that need help to get through, they can get through as they need to, okay? The only other kind of transport that we talk about is active transport. Active, because it's active, meaning energy equals ATP. You gotta have it. You gotta have ATP energy to do this, okay? So for active transport ATP, in this case, we're talking about pumps, stuff like that. The two things that we kind of talked about were when you take in bulk stuff through what we call endo or exocytosis. When you push things out or you bring things in. Exo, you got to exit. Endo, where am I going? I'm going endo. So you're going inside. The type of endos that we talked about, phago means to eat. So your cell is eating something. That would be a lysosome eating a bacteria, or sorry, the bacteria eating so it can take that lysosome and communicate it with the backup. The cell eats the bacteria, pushes that bacteria to a lysosome so the lysosome can kill it. And the other one would be Pinot. Pinot Noir. It's wine. Why? Pinot means to drink. Okay? So you either eat or you can drink. But again, this is all active. It all requires energy on the cell's part for it to do that. Okay. Okay. Dang it. My thing's kind of stalling real quick. Let me make sure I can get up there. Okay. So Rachel, let me know if I need to go over that anymore with you. Quick overview of cell respiration and photosynthesis. Okay. Hunter Lawson. Who's Hunter Lawson? Hunter, let me T-pose on the head of the star. I don't even know what any of that means. Hunter, oh, Hunter, is that you? And I'm just seeing like your middle name or something? Okay, quick overview of cell respiration photosynthesis. Whoo, Katie, how much more do you want me to go into? I kind of did the Krebs cycle and Calvin already. Overview of cell respiration and photosynthesis. It helps if you just break them up into just kind of a quick little chart. So cell respiration, photosynthesis. First thing you want to do is where do they take place? So cell respiration, mitochondria versus the chloroplasts. In the mitochondria, the first stage is glycolysis, which is the breakdown of sugar. You can see because it has glyco for glycogen, lysis for break. First stage over here would be the light-dependent reaction, okay? Using light to help break down water. Where does this take place? The where over here in glycolysis is the cytoplasm of a cell. The where for the light dependent is the thylakoid inside the chloroplast. Okay? Step 2 in a cell respiration would be it chooses, is it going to stay in the cytoplasm and go through fermentation, or is it going to go into the mitochondria so that it can go through actual aerobic respiration? So we're going to go ahead and choose aerobic, so it gets to move into the mitochondria. So the second step would be the Krebs cycle, and that's taking place in the matrix, the open space. Step 2 over here for the light-dependent reaction. After it does all that processing inside the thylakoid, also part of the ETC, the second stage it's going to go into is the light-independent reaction, which happens in the stroma, which is simply just the Calvin cycle, open space, open space. And then over here, after the Krebs cycle, we go into the last stage in cell respiration, which is the ETC, which happens in the membrane of the mitochondria called a cristae. Over here in the chloroplast, that's all we needed to do. We go to the thylakoid, we go to the ETC, make energy, we go into the stroma, and we make sugar. Over here in the mitochondria, we break sugar, we made some energy. Krebs cycle, it makes CO2 so we can breathe it back out. And the ETC helps us make our water so we can urinate it out. Okay, kind of a quick little overview. Decomposer, dear Lord, if I need to tell you what a decomposer is, dear Lord. We got a group kid in here. Oh, is that what that is? Okay. Oh, Hunter. Duh. Hunter. Oh, gosh, that just now clicked for me. Okay. Hey, Hunter. May I ask questions even though I'm in Morris's class? Yes, sweetie. Go ahead and ask your questions. That's fine. Can we get a quick T-pose? I don't even know what you're saying, T-pose. What do you mean by T-pose? What is a T-pose? I have no idea. Sorry, I'm having to go through all of your questions. They all literally just popped up. Quick explanation of ecological succession. Okay, I like Bob. I'm glad you like Bob. Okay. All right, hold on. Okay, ecological succession. Guys, there's only 2 kinds of successions, okay, that we discuss. Yes, there's actually a couple of more, but really it only comes down to 2. Primary and a secondary. Primary means it's the first time you've ever seen any plant life there. That's primary. And primary is the only time you can see a pioneer species, okay? And that's lichens. Lichens are simply just a fungus and an algae. They help break down the rock so that it can eventually become soil. And, oh, now you got plant life, okay? Secondary succession, look at it. It says secondary, meaning something's already been there first. So secondary happens on soil. So when will you see either of these occur? Primary, if a glacier is receding and you're starting to see ground for the first time, it's going to be rock. If a volcano is erupting, like the one erupting over in Hawaii, one of the islands right now, crazy, it's destroying everything, okay? Secondary succession would end up, being a farmland. They end up destroying the farm, right? They've got to break it all down. Why? Because, well, yeah, it's actually a good thing, right? To actually kill off the crops, release all the nutrients back into the soil. Secondary successions also happen after some natural disasters, floods, fires, that kind of thing. All right. Hello, Daddy Slate. Hey, okay. I'm going to have to, okay, I don't know what that is. Can you go through the where you said to draw a cell in a cup and that kind of thing? So that's going to be your hyper and hypo and isotonic, okay? And I'm hoping my Wi-Fi is working pretty well. Like I hope there's not a whole lot of glitch kind of stuff, okay? Oh, yeah, Twilight, yes. Thank you, Carson. Hunter, Twilight, totally. Sorry, it's all coming back to me right now. I guess, Hunter, I'm not used to seeing your last name, so it threw me off for a sec. Okay, redhead who walks into class with me, yes. Who is Twilight. I'm sorry, but Hunter, you're just always going to be Twilight to me. All right, when senior year, I'll like sign your name somewhere and I'll put Twilight. Okay, the solute. So a question might ask, let me just kind of write this down. A paramecium, okay, lives in fresh water. We know paramecium live in fresh water because they are a protist. Even if I didn't write that, you should be able to tell me a paramecium lives in fresh water because protists are found in pond water for most of the part. Okay, so a paramecium lives in fresh water. You place the paramecium in distilled water. Okay. How will the water move? So you took a freshwater organism and put it in distilled water. So on any of these questions where it's talking about water moving and it's having to deal with a cell, what I suggest is, again, draw it. So I'm drawing a beaker and I'm putting my cell in it. Nothing special. I put it in a beaker full of distilled water. The percent we are concerned with are for salts. Because it's distilled water, we have 0% salt. There is no other ions in distilled water, nothing. So right there, if that's 0%, we can't get less than that. So since I don't have a lot of salt, our prefix suffix terms we know. Hyper means more, hypo means low, and iso means equal. So to describe 0%, I definitely do not want to use hyper. I would use hypo. So the solution is hypotonic, low salt. That means that I get to label the cell hypertonic. Water loves salt. Water will go where there is more salt. So the word hyper means more. So water will go where more salt is, hypertonic. So your only options are into or out of the cell. Since hyper is in the cell, water is going to rush into the cell. The cell is going to soak up water like a sponge. Because this is an animal cell, if it takes in too much water, what could potentially happen to a cell? Burst! Ah! Death of the cell! It can lyse. A plant cell won't. A plant cell loves to be in distilled water. Ooh, as long as you give it plenty of nutrients for the other stuff, it loves distilled water because it's going to swell and push on its cell wall and make it turgid. Okay, if you need another one, I can give you another one. Peyton, just let me know. Okay, I'm just going to try and answer some other questions so I can catch up. Hypo or hypertonic, Cass, so are you. I don't even know what any of this. May I get a T-pose? I still don't know what a T-pose is. I would like a T-pose on all the biologists. Okay, you realize that I'm going to look up whatever this T-pose stuff is, right? Like, I'm going to look up whatever this T-pose is. I know that. I would like to be, okay. Hey, Miss Good, it's Charlie's, so I forgot which is on the bottom of the food chain. That would be your producers, all food chains. So if you're drawing an ecological pyramid, the bottom of your pyramid must be the largest because it needs to support the rest of your pyramid. So that means producers. That means any kind of plant, bacteria, or algae. And then you go into a primary consumer, secondary consumer, and then tertiary consumers and on up there. Okay, 110, 1.1, always 10%. Okay, notice who's not drawn on here. But the circle of life has to always end with them. Not drawn on there would be your decomposers. Okay, you can't forget the decomposers on this. They're just usually not pictured on here. All right, producer, sorry, people already answered you. Are you sure? Yeah. I want to make sure, so let her answer. Okay. Charlie, it's simple producers. Da, 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 da. Okay. Ah! A lot of stuff just like popped up, so I'm having to like play catch up here real quick. Okay. Can you go over cladograms with dates like the one on the STAAR review packet? Okay, I can try to real quick, but man, that one was definitely more difficult. But here's the thing. It was only difficult because they threw numbers on there. But really, all you need to do is draw it like a timeline. So I'll try and show you what you can do. Okay. I do not know what a T-pose. I don't know what you're saying. According to all known laws of aviation, there's no way a bee should be able to fly. Thank you. I appreciate that. Okay. Because bees, I don't care about that according to, okay, don't care. Charlie, I already got you. Okay. Yeah, sharp. All right, some of y'all with all this other crap, eventually, if you're going to keep doing all of that, I'm just going to kick you out because I actually can kick you out. Okay. Because some of you that are putting all this other crap on here, you're making it difficult for me to actually help some of the people that are actually on here. Don't make me waste my time. All right, so let me look at a cladogram, Heather. So I'll do that for you. And then I see another question that says DNA replication. So cladogram, DNA replication, and meiosis. Okay, DNA replication and meiosis. All right, what I can tell right now, it looks like Jefferson, if that's you, I'm about to kick you out. And I'm about to kick out cool person, okay, because y'all are making this really difficult for me to read. Okay, cladogram, DNA replication, meiosis. Those are the next few things. So that cladogram on the star, so it had all this, and it had things like green algae down here, okay? Then it had, I don't know what some of them, how it had it in order, but it had like moss, club moss, ferns, and then, you know, angiosperms and stuff. But instead of having a regular characteristic, it had the characteristic like vascular tissue, but then it had a date, like 409 million years ago. So that kind of tripped y'all up because the question ended up asking is, which of these discoveries, so they were, scientists found something, does not provide validity for this picture. So basically saying, which of those doesn't match the others? And that's kind of how you can always attack any of these questions. Even if you're not sure what the question's asking, just read the questions and see which one doesn't, like which one are you not saying yes to? Which one are you saying no to? Which one does not match the rest? So on that question, what it ended up saying was, so one of them said, you know, a scientist found a green algae dated 100 million years. So the best way you could kind of redo this was draw it out like a timeline and put, okay, vascular tissue, 409 years ago. The first fern, 200 million years ago. Did it, you know, stuff like that, okay? Make it into a timeline. That's why that would help because the questions were based on that. So if I found, so let's say, you know, all the way back here is the green algae and there was something else here. So one of them said, you know, it said a scientist found a green algae dated 200 million years. Well, if green algae were back here 500 years ago, could a person go pick up algae that was only 200 years? Well, yes, you can go pick up algae right now. So yeah, that is reasonable, okay? Another one said that they found a vascular plant dated 300 years ago. So about right there. Well, vascular tissue occurred here. So could you pick a vascular plant 300 years ago? Yeah, because that happened after it. Check. But the one that stood out said something like, you found a fern which doesn't exist until here, 200, but you found a fern dated like 400 years ago. What? It didn't even happen. Ferns didn't even exist till 200 years. But you're saying you found one at 400 years? Something's weird about that. So that's the one that you say, oh, no to, okay? Okay, replication. For DNA replication, that's what we did on the board. You have a 5 to a 3 of DNA. So if one side of DNA said A, T, G, C, A, T, then the other side of your DNA, when it's replicated, has to be the opposite. A 5 must always pair with a 3, and a 3 must always pair with 5. Apples grow on trees. Cars go in garages. Apples grow on trees. Cars go in garages. That would be simple DNA replication, right? But I think what you're also asking is in the cell cycle. So actually, let me go ahead and draw this. DNA replication in the cell cycle means during synthesis, right? So in your cells, the first picture of cell would be indistinguishable. You couldn't tell anything in the nucleus. Why? Because what's happening in there is DNA is being replicated. That's during synthesis. Every chromosome gets another copy of itself. Okay. Now, all the enzymes, DNA polymerase, DNA helicase, ligase, really the only one that they may even ask is helicase. There were 3 enzymes we talked about. When DNA's double helix needs to be broken so we can copy it. So we can make a new strand here and we can make a new strand there. Okay. You had to split DNA first. Well, to split the DNA strands, you have to use helicase because H for hydrogen bonds. To add new strands of DNA, you had to use polymerase. Why? Because look at its word. Polymer. You got to build the new DNA polymer. And then to glue the pieces back together, you had to use ligase. Why? Because ligase links the pieces. Now, what pieces will you see ligase linking back together? Okazaki fragments. Okay? Those were the lagging strand because remember, you can only make one strand continuous, but then the other strand has to actually be made backwards. Okay? Using Okazaki fragments. So the only thing that you really would need to know for DNA replication would be, the polymerase builds because it's a polymer, helicase breaks hydrogen bonds because they're weak, and ligase links those fragments. Okay. Kayla, I will do meiosis for you in just a second. Let me catch up on some of these. Are we okay if we know everything you talked about in class, or do we also need to go a good amount specific details? Joe, if you kind of already know what we did in class, you're pretty much good. What y'all are going to find is there's going to be specific details that you're going to be like, wait, what? Yeah, because those are probably the free response stuff. How do you know what the levels of... Okay, so my question is, how do you know what's the levels of the food chain? I'm not really sure what you're asking. I think you're just asking for the food chain levels, which again are your pyramid, trophic levels. You go from a producer to a primary to a secondary to a tertiary. You always have to start with a producer. Okay. Okay, so my question is how... Okay, you already said that. Okay. Okay. I'm deleting some of y'all. Okay, I got it, Miss Good. Glad that you showed me an example of distilled water. Okay, good deal. Okay, look it up. It is up to you. All right. Sorry, I'm sitting here just trying to look at some of y'all's stuff. Okay. Sorry, some of y'all I'm kicking out, just so you know. So if all of a sudden you're kicked out, it's because you're spamming crap and I need you to get out. Okay. DNA replication. I don't know Allie. Oh, Allie. Oh, Allie. It says Allie Henry. But okay. DNA replication I already kind of did. So yeah, hopefully that... Cool Stop Kid. Jeffy J, I am kicking you out. I am thinking that is you, Jefferson. So if that is, I'll give you a second. But I'm about to kick you out too. All right. So Michaela, okay. Michaela Fritz, you're a very cool monkey in the jungle. All right. I don't know who Michaela is. I'm guessing you're Michaela from class. All right, Miss Good, what's a good way to remember the difference between carbohydrates and lipids? Just shape. Carbohydrates are always going to be hexagons or pentagons. Lipids are always going to be straight chains that you're going to see like at the end of a phospholipid. Okay. If some of y'all know who I need to kick out, who's not legit, just tell me and I'll kick them out. Okay. Can you do the ones where it's talking about the pairs and it can have deletion, substitution, stuff like that? Yep. All right. So Kayla, you wanted some meiosis. Let me go ahead and give you that real quick. So remember meiosis, that's for sex cells only. The big issue with the big thing you got to remember with meiosis is you start with the diploid, 2N. Okay. Meiosis goes through P-mat twice. So it goes, meiosis does I, P, mat, C. And then it does P, mat, C again. There's meiosis 1 and meiosis 2. So you start out with 2N. What that means is you have chromosomes that have their homologue. So if I count, there's actually only 2 chromosomes here, but there's 4 chromatids. They split up and you immediately go to haploid. Okay. So at this point, you have this chromosome and that chromosome separated from each other. Then they go to haploid again. N, N, N. Because they split from their chromatids. Okay. So you went from 2 to 1 to 1 in each of these. Meiosis' job is to make 4 haploid cells. The purpose of mitosis is to make 4 diploid cells. We call them daughter cells at the end. Okay, deletion, mutations, substitutions. Okay, so anytime you have a mutation, you're talking about a mutation inside your DNA strand. That's where we're looking. It's not a mutation in mRNA. It's a mutation in the DNA. All right, so if my original strand of DNA said this, A, T, T, C, C, G, A. If my original said this, and they said, which of the following examples shows a point mutation? First of all, if it says a point mutation, that has to only be one, and it can only be a substitution. Points can only be substitutions. Okay, so in this case, my answer choices would be A, T, A, C, C, G, A. T, T, C, C, G, A. A, T, 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 C, C, G, A. So in this case, if I gave you those 3 options, the only one that can be a point mutation would be the first one, because I changed that second T to an A. Substitutions, they have to be the same numbers. If it's a deletion or an insertion, it's a frame shift. Okay? Because I've actually changed how I'm reading it differently. An insertion or deletion changes how many numbers. So I got rid of my A here and I added a T here. Okay? So DNA replication happens in the S phase, yes. Okay. All right. Some of y'all on. I'm trying to, that's my mom's maiden name. Okay. Okay, kicking a few people out. Okay. And so I'm trying to kick some people out. Jeffy, Feely. Okay. Okay, I don't know which ones are which. Okay, is it kings play cards on fuzzy green stools? Yes. Okay. Fuzzy green stools, not chairs. Okay, kings play cards on fuzzy green stools. And really, you can add in the D. So dumb kings play cards on fuzzy green stools. Dude, you just spammed hypocrite. Okay, I don't know. As long as I can get through your questions. Yep, don't forget the domain. You have 3 domains. You got to talk about archaea, bacteria, and eukarya. And then you can talk about all the kingdoms and the phylums. And you need to know in inclusive versus exclusive kind of thing. Okay. Bro, no, don't kick. Okay, some of y'all getting kicked out again. Okay. What's the passing rate? As far as my class goes, 100%. As far as our school, again, almost 100%. We usually only have 1, maybe 2 people in our entire freshman classes that don't pass the STAAR biology. Y'all should do just fine. I personally, for my classes, I want to see 60% mastery from my students. But again, almost everybody does. Are they going to ask about dinoflagellates and are those protist groups? They are protists. It's rare. What you would actually see is they're going to give you a really bad, blurry picture of a protist, okay? But you don't know it's a protist, okay? They're going to give you some really, really bad, very blurry, you can't tell anything going on kind of picture. And they're going to give you a description about it. Or they're going to give you a dichotomous key to go about it. And you need to determine if it's a protist. First of all, if you're looking at something like this and you can clearly see a nucleus, you can X out anything that's a bacteria. Okay. Second thing, how many cells do you see that this is being made up of? It looks like it's only 1 cell. So you can probably knock out animals, probably. Okay. And then if it says anything about it being in pond water, ding, 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 you know it's a protist. As far as them asking very, very specific, like dinoflagellates and what kinds of algae and stuff, no. The only one that I've seen really are diatoms. Remember, diatoms are the special ones that have glass cell walls. Okay, do we have to know all kingdoms? Well, yeah, there's only 6. Think about all the things in your world. You've got you, an animal, plants, fungi, protists. We talked about all those, right? And then you've got your 2 bacteria kingdoms. Archaic ones that are extreme lovers. You don't come in contact with those. And then your eubacteria, which are your everyday guys that live in and on you, help you or hurt you. I think you have to get like 25 questions right. Really, technically, yeah, to pass, that's all you have to. Okay. Cast, they're protists, not fungi. If I don't, whatever. What's the score you have to get to pass? Again, I think it's like 25 out of 54 this year. Okay. Also, how many do we need to get right? Okay, well, I already answered that. You have to get like 25. What is the score we made on the practice say about the score on the real STAAR test? Pretty good. That was a STAAR test from 2014. I didn't print off the most recent one because that's just what I had available at the time. But it's pretty good. It's whatever. In also reality, if you're in my pre-AP class, there's no reason why you shouldn't be getting mastery on it unless you've just been totally failing and skating by in my class. But yeah, if you really, I don't know whether or not you really did that test for real. If you cheated, if you looked up answers, if you talked to people, I don't know. But you know if you did. If you legit took the practice STAAR last week, you didn't do anything weird or crazy, and you took it and you got 30 plus out of 54 right, then you're just fine for tomorrow. You're perfect. Okay. If you got higher than that, if somehow you actually ended up getting, like 46, then you're looking at mastery tomorrow. Okay. All right. I'm going to go through the packet I've got at home from the 2014 one. That would be good. Guys, none of this STAAR is hidden. All you have to do is look up biology STAAR and it's on the TEA website. Is energy the same thing as heat in a food chain? No, but it transfers differently. Okay. So we transfer energy 10% throughout the trophic levels. We lose some of our energy to heat in each trophic level. That's why only 10% gets to be transferred. The other 90% we're using for ourselves, our metabolism, our heat loss, our food consumption, our feces, that kind of thing. That's why only 10% is available to the next energy level. Okay. All right. What else? Will, I'm about to kick you out again. All right. I think I kind of hit on all the questions after I got through a bunch of that spam crap. So I'm kind of at the bottom. If I didn't hit your question, you can probably go ahead and ask it right now, and I might be able to kind of keep up. I'll stay on for probably another 10, 15 minutes, but I know I want to go to bed at some point. I have crap I need to do. But if there's some stuff you need, obviously go ahead and say something so I can see it. Okay, Hunter, can't really give you a quick summary of the biology at this point. Can't really do one. All right. Water is a polar molecule. Yes. Water is polar because polar bears swim in the water. You have to know that. So only things that are polar can actually mix with water. Things that are non-polar like oils and lipids can't. Okay. Dihybrid. So you have to know the individuals. If I said an individual is hetero, but then homodominant, crossed with a homorecessive hetero individual. So Cass, while I start looking at some of these questions, go ahead and write that down since you asked for this. Okay. Can you go over the Punnett square with two traits? That's what I'm doing right now, Abby. So is it G1 synthesis and then G2? Yes. My mom heard you say crap. I don't know when I said that. Sorry. Hi. Mama Creesh, how are you doing? Okay. I really don't even know when I would have said that. Okay. Let's see. All right. Give you a second to kind of write that down. Then I'm going to show you. On a dihybrid, okay, you want to look at the letters individually. What snacks do I recommend for brain food? Anything at this point. Okay. So I'm going to look at the A's, and then I'm going to look at the B's only. So do your normal Punnett square like you always would. Fill that in. The first one, I get 1/2, 1/2. GG, I'm grounded. You didn't say it. I said it, right? Girls do not have the XY. Girls keep their legs crossed. Okay, XX. Boys have the XY. Okay. Yes, she is. She is not going to ground you. What group of organelles is directly responsible for the production of new molecules within the cell? Okay. Hold on. Okay, what molecule? It says what group of organelles is responsible for the production of new molecules within the cell? I don't know if that's the whole question or not. Okay. All right. WAPAP, I'm about to kick you out. All right. Boys have Y. Think about it's Y, 2 testes, and oh, dear Lord, Charlie. Okay. Cass, the what group of organelles is directly responsible for the production of new molecules within the cell? In that question, I don't know which one it's like how you're referring. If you're looking at the test question, I'd have to double check it and see what answer choices it gives you, because the nucleus itself is what's helping to produce a lot of stuff. But then the other question it's probably going to help is the Golgi apparatus, because without the Golgi, nothing can actually be pushed out. Okay. All right. So on the dihybrid, go ahead and do the Punnett square for the B's. As long as you look at them as individual letters, that helps you. And then we have to do the fractions. So when you complete the second one, you see that it's 100% dominant. So write it as a fraction. 2 out of 4 are capital A, and 2 out of 4 are recessive A. 4 out of 4 over here are capital B. So you need to find all the possible combinations. Capital A, capital B would give us a 2/4 times a 4/4. Okay. So 8 out of 16. In this case, lowercase a, capital B, would give us, again, 2/4 times 4/4, 8 out of 16. Notice those are the only combinations I can do. One, because 8 and 8 equals our 16, but also I can't do anything else because there are no other combinations for B that I can do. How much is this going longer till? Only for about another 10, 15 minutes, whatever y'all need. But at some point I am going to say we're done. Okay, what are the products and reactants for respiration? Yeah, Charlie, you might want to retract that crap. Respiration, I'll answer that. What are the types of different fibers? You don't need to know that, Joe. Is 2 to 7 days lytic? Yes. That would be a fast reaction. Okay. Now, to finish this up, you would simply just say 8 or 50% would have dominant, 50% would have recessive dominant, and you would be done. Okay, reactants and products for respiration, you just have to know at this point. You're going to have to just know the equations. So the best way to attack it is think about what a plant needs to survive. Okay, a plant needs water to survive. A plant needs CO2 to survive because they take in our atmosphere. And then a plant will produce glucose for themselves, C6H12O6, and they will produce oxygen for us. If you can remember the plant one, then you know the respiration one because it's just reverse. Reactants have to come first because you have to react things to make a product. Okay, quick overview of evolution. We'll see if we're understanding all this. You think we'll be okay? Yes, you're good. No, I didn't. Saying boo-hoo, scary poo. Okay, I don't know what you're saying. Can you kick out Charlie? Yes, I can. Okay. Lytic is short. Yes, Eli. Lytic is shorter than lyso. Okay. Yeah, Carson, you're fine. Go ahead and get off if you want to. Evolution, I'll look at in just a second. Wheel of formation. Okay, you are gone. Okay. All right. All right, so what was that next one? All right. What are ions? So let me look at the first one. Somebody just, I forgot what I was just about to look at. Evolution and ions. Evolution and ions. So let me answer that real quick. All right. As far as evolution, I can't really give you a quick overview of it, but just remember natural selection occurs because organisms have better adaptations than others. So if you have the best adaptation, you will be selected by nature to continue to pass on your genes so that your best genes can then follow into your offspring, leaving you to be the most fit organism. Remember, fitness means you were picked and you have and you can reproduce. If you can't reproduce, you're not considered to be a fit organism. Things involved with evolution, you need to remember things like the Hardy-Weinberg, right? And we did the gene flow, genetic drift, we already talked about at the very beginning. And just the fact that evolution occurs through allele frequency changes. Ions, yep, the person already answered that for you. You have either cat versus Ann. Remember, plussy cat, negative Annie. So if you know those, then that's all you would need to know. I doubt that any kind of question over ions would actually be asked, though. Okay, highly, highly doubt it. All right, anything else? Mitosis. I pee on Matt C. Yes. I pee on Matt C. Correct. So, like when you get your test, thanks for saying bye, back, but bye. Okay, can you just show us an amino acid chart? not the pi one. I did in class. I can't pull one up because I don't have one physically in front of me, but you read it the same. On any of the charts, you would read it. So if the chart had it like this, you would read this side first, then this side. Find the box where they meet. And then in that row, you would find the last letter. And then that would be the amino acid in the box. Okay. But yeah, I can't really show you one because I don't have one physically in front of me. What is IP on Mat C? I'm in Morse, and he didn't go over that. Oh, okay, Zach. IP on Mat C is just your phases of your cell cycle. It's just a sentence that we use to help you remember it. So if you can remember the sentence, and as soon as y'all get y'all's test, I'm not kidding, as soon as you're able to write on it, go ahead and throw down on the first back of the page all these acronyms, all these pictures, everything you can remember, right? The the King's Play cards on fuzzy green stools, the IP on Mat C. Start to draw out a Punnett square, write out the photosynthesis equation, whatever you can. Diploid, haploid, 46, 23. So IP on Mat C, Zach, means interphase, prophase, metaphase, anaphase, telophase, cytokinesis. We remember it as a sentence. We insert the word on, Mat C, IP on Mat C. It's a way to remember the cell cycle in order. Also to remember is your hand signals. Out of these 5, if these 5 fingers represented chromosomes, they would hide inside of my nucleus. So you can't see my chromosomes right now. They would then be synthesized. So I actually have 10, but again, they're hidden. So this is still I, synthesis. P, I can see them now because in prophase, the nuclear membrane disappears. So I can see my chromosomes now. M, they line up in the middle, right? M's the middle. So see how they all are connected as X's in the center, okay? A, they get pulled apart. And at the same time, they're screaming, ah! Why? Because these sister chromatids that were replicated are now being pulled apart. So A, they get pulled apart. T, they go hiding into their nuclei. Cytokinesis, they separate. Okay. I, synthesis. P, M. A, T, cytokinesis. Now, the main part about doing this is seeing that you started off with one cell that had 5 chromosomes. At the end, you ended with 2 cells that each have 5 chromosomes. That's the purpose of mitosis, to end with daughter cells that have the same number as the beginning. Okay, can you show the hand motions? Just did that. In mitosis, it isn't only PMAT division. The whole thing is, okay, so what I just said, IPMAT C, that is the cell cycle. PMAT is mitosis because that's the division of the nucleus. Inherited variation, Cass, all that's literally meaning is the genes that are being passed on, that you're inheriting the different versions of alleles. What if one were to fail mitosis? I don't know what you mean. Is the stomach acidic? Yes, you have hydrochloric acid in it. Okay. Let me see. All right. Okay. I finished going through the packet, so I should be good, but I'll stay till the end. Some of y'all just hid because you're cluttering up crap. What about benign tumors and stuff like that? Very doubtful. The only thing that would be asked is just what is cancer, okay? Cancer itself is just mitosis uncontrolled. It's the cell cycle that has not stopped, and so it's your cell's body kind of just doing it to itself. That's all that would be asked about cancer. What if someone only has 1 kidney? Why is that? Doesn't matter. I just learned more on this live stream than I have all year. Good. Yes, tumors are just clusters of cancer cells. Okay. Right. What else? So we kind of did biochem, did some chemistry, did some respiration and stuff. Genetic engineering won't be on there, right? So my classes, when we talked about gel electrophoresis and everything and that kind of stuff, none of that would be on there. Okay. Okay. Is there anything they ask a lot of? Cass, it totally depends. Every year, it seems like they choose to kind of focus on some stuff more than others. I know one year it was like so ecology heavy, and then the next year was evolution heavy. So we never actually know. So they say that they include this many questions in this and this. They could ask tons of questions on evolution, but because in those evolution questions they said a biochem thing, they get to claim it as biochem. So it's totally dumb. Okay. What else? Anything else I could potentially answer for you guys? All right. I have no idea what that is. I'm not looking up T-pose. You're welcome, Joe. Will our year be over after this test? No, we do dissections. I have no muscles to flex. What is the photosynthesis, Brian? We did this earlier in the video. To know the photosynthesis equation, you just need to think, what does a plant need to survive? Okay. A plant, yes, we're dissecting. A plant needs air, CO2 to survive. It needs water, it needs sunlight. That's its reactants. Then a plant will make oxygen for us and glucose for them. Those are its products. Okay. Yeah, kind of fun stuff. Thanks, Miss Good. You helped me. You're welcome. I love internal organs. I do too. Okay. I'm still here if y'all have anything else, or if some people are just now kind of getting on. If you're banned and you're just now getting home, the live stream is still available. So you can just, whenever I go to end it, you can just click on it and watch it again. Will, I'm not sticking out my arms and doing T-pose. All right. Anything else? Ban people, just feel free to watch this whenever you need to. Oh, the only other thing that I might mention is a karyotype, right? I doubt it'd be on there, but might as well go ahead and mention it. Karyotypes, those are the pictures of your chromosomes, right? So you got the chromosomes, and they're all in order. Chromosome 1, 2, 3, until you get to the very last ones, which are always going to be your sex chromosomes. First of all, in a karyotype, that's all it is. A picture of chromosomes to determine whether or not the person is genetically healthy. If it's human, you should have 46 total. These last two are sex. If they are two different sizes, that means it's an X and a Y, because the Y is junk DNA, so it's very small. If they're both relatively the same size, then they're both X's. You're looking at a female. Okay? And when you're looking at the karyotype itself, you should only have 2 per numbering. Because if you see a third or if you see a single, that means the person is unhealthy genetically. Okay, pedigrees. Allie, I already kind of looked at pedigrees earlier, so you can just kind of watch the video from prior. Pedigrees, we did that in class as well. Circles are always Mrs. Puff, right? So Mrs. Puff is the circle. SpongeBob SquarePants, he's the male square. Shading means that they have the trait. Yes, if there are 321s, then yes, the individual has Down syndrome. Okay. Weird, Hunter. Weird. Will they be half shaded on this test? No, doubt it. But even if they were, half shading means that they carry it, which means it's recessive, and they are heterozygous because they don't express it, but they can carry the trait. Carriers only exist on recessive. Why do I need to T-pose for them? It's T-pose. All right. Anything else I can do? All right. Anything for you guys? Bye-bye. Will do, Cass. They're all in bed. Biochem before my test tomorrow. Come to the lecture hall and T-pose, dear Hunter. I'm in the lecture hall tomorrow. Okay, biochem, Will, if you really need it, I'll go over it for just a quick sec. Okay. Just a quick sec. Will, do you actually need biochem? Okay. Biochem stuff. Okay. There are only 4 molecules that we're concerned with. Carbs, lipids, nucleic acids, I'm just going to put in A, and proteins. The reason we're concerned with all of those is because all of those are organic. The only reason they're considered organic is because they have carbon in them, no matter what. You are an organic being because you have carbon, okay? Why do you get the boys? I wish I had you in my room. I don't know why she puts me with the boys. I have no idea. Okay. All right. So they're all organic. They are all carbon. All of these are polymers made up of specific monomers. Unique things you should remember. Carbs are either hexagon or they're pentagon, okay, in their shape. Lipids are either going to be saturated or unsaturated, and they just look like chains. Nucleic acids, you should always recognize the nucleotide shape, PBS. You know that that's an S because it's a pentagon. You know that's a P because it's a circle. And then proteins, you should always remember that the bond that bonds them are peptide bonds. Okay, it's kind of the important thing. Also in biochem, you look at a phospholipid, right? We talked about this in class. You know that this is a phosphate because it's a circle. The head versus the lipid tails, because those are lipids, oil doesn't mix with water. So lipids are nonpolar and they fear water, so they're hydrophobic. While the head is polar, it likes water, so it mixes. So when you see a phospholipid bilayer, the tails are stuck on the inside of the membrane, while the heads are exposed, because this is the outside of the cell and this would be the inside of the cell. Okay. All right. Non-Mendel. Okay. Well, what else on biochem do you need, Will? Like what other stuff besides just monomers, polymers, and stuff? Like we already kind of looked at enzymes earlier in class. I don't know what others. I think I'm good. All right. Non-Mendel, Will. All non-Mendel was either co-dominance or incomplete dominance, okay? So if it's co's your prefix word for together. So because they're co-dominant, that means they are both there. So a co-dominance, that's your dairy cows, that's your Dalmatians, that's roan-colored horses, that's black-peppered chickens. Anytime you see 2 phenotypes at the same time, that's co-dominance. If you look at something and you can't actually see their phenotype, or you can't see the original parents, is what I meant, then it's an incomplete, because incomplete's a blend. Because think ink for pink, okay? Because that would be red and the white snapdragons. If you were to mate them together, you get pink flowers. So you don't see red nor white in the offspring if it's an incomplete. Yeah, if you've been getting A's on my test, you're going to be just fine. Like I said, if you did that STAAR legit, then you'll be fine. Oh, yes, he did, Mendel. Also, you can give me my scores. Yeah, Cass, I'll send it here in a sec. Okay. All you needed to know about Mendel was being able to do Punnett squares and knowing genotype and phenotypes. Okay. All right. Anything else? You're welcome. Could you also send my practice STAAR rescores? Yes, I'll go through the ones that I didn't and I'll send them back out. All right. Anybody else need something? Merci? Anything else? If not, then we're going to say we're wrapping up. And if you need to, look at this again. Can you explain what the organic compounds are composed of? Organic compounds just means that they're composed of carbon. And just literally 5 minutes back in this video, I quickly went over the carbohydrates, lipids, proteins, nucleic acids. Those are your organic compounds that we talk about from biochem. So if you remember their shape, you remember how they interact, then you're good on that part, Lily. Library is always closed on STAAR days because some students have to be in there. So no. Anything else? Sorry. No books. I don't think I have anything interesting. All right. Give you about another 5 minutes. See if there's anything else that I can answer. Other than that, Lily, you might have just gotten on, but I said band people, y'all might just want to look and watch this video again. Okay. Just so you can kind of go through the stuff that we discussed. All right. Thumbs up. All right. Good thing, guys. I'll see you in the morning. Get some rest, eat something decent, bring water, bring something to read afterwards, and you'll do just fine. All right. Bye.